Good afternoon and welcome to the 2022 Entrepreneur of the Year presentation. This is part of our annual UMB Founders Week. This award is named in honor of Dr. David J. Ramsey, former president of UMB. Dr. Ramsey was a fearless advocate of entrepreneurism and was very much the force behind the creation of the University of Maryland, Baltimore Biopark. His creativity and persistence on this campus lives even today through this award. The process that we follow is that we receive nominations both from within UMB and also from the external leaders in this community. And this award then is based upon entrepreneurial activities brought out by those consultations. And basically we base that upon the creativity and importance of the work being done, the potential impact on society, the success at overcoming obstacles, of which there are always many obstacles, and lastly, the individual's collaboration with other non-academic entities, such as governmental agencies, private businesses, foundations, etc. So I thank you for being here today for this award. I could not be here personally to thank Dr. Allan Doctor and congratulate him on this award, so let me do that now. Allan, it's a great accomplishment. Since the day you walked into UMB, your presence has been felt, your deep research has been appreciated, and your commitment to entrepreneurial activities is always palpable. Congratulations on this award. So with that, I would like to turn this over to Dr. Mark Gladwin, Dean of the School of Medicine, who will further describe Dr. Doctor to you. Thank you.
Thank you. Well, it's a real great honor and pleasure to introduce Dr. Doctor to all of you. He and I share many passions in science and medicine, passions for nitric oxide biology and signaling, hemoglobin, sickle cell disease, red blood cells, hemoglobin-based oxygen carriers. And I'm particularly jealous of the beautiful lab space that he has. And I now have made it. It's one of the shock and awe experiences of my first 2 months here, seeing that lab space. And as he'll share with you, every visiting dignitary I send to see that space as well. So congratulations on that as well. By way of formal introduction, Dr. Doctor earned his bachelor's degree in neurobiology and his medical degree from the University of Virginia. He completed a residency in emergency medicine at the University of Pittsburgh. He went on to Boston Children's Hospital and completed additional residency training in pediatrics, as well as fellowships in pediatric emergency medicine and critical care medicine. He then went to Washington University in St. Louis, at St. Louis Children's Hospital, and served for 10 years there as the pediatric critical care in the division and as the division chief of that division. And then he was recruited here to lead this remarkable program that you're going to hear about. His laboratory studies the role of cell-based signaling in red blood cells. He studies how red cells can control regional blood flow, how red cells modulate pathophysiology after red cell injuries like hemolytic conditions. He studies the role of red cells and hemoglobin in blood substitute therapy design, and the role of red cells in transfusion medicine and critical illness. He's published more than 180 manuscripts, has an H index above 50, so that means there's 50 papers that have been cited more than 50 times. Very remarkable. He's been cited more than 10,000 times, and he has 11 patents related to his work. His research program has been robustly funded by multiple awards from the NIH, the Department of Defense, and private foundations. In particular, he's a PI for a 14-site consortium based here at the University of Maryland School of Medicine. It's recently been selected for a $38 million award from DARPA. The project is titled CONCERT, Consortium for Optimized Integration of Bio-Artificial Blood Components for Adaptive Resuscitation and Therapy. And the award is in final contracting and expected to commence in January of 2023. He directs the School of Medicine Center for Blood Oxygen Transport and Hemostasis, which addresses fundamental challenging questions related to blood oxygen transport and hemostasis, that's clotting. And it spans the translational spectrum from human biology to therapeutics. Specifically, they've launched several therapeutic development projects that bridge academic discovery science, pragmatic pharmaceutical and bioengineering, and ultimately attempt to translate these things to the clinic. One example of one of the projects is the use of artificial intelligence and machine learning to identify pre-organ failure physiologic signatures of anemia, so that to figure out who would need to receive transfusions, who may need novel therapies for sickle cell, treatments to mitigate hemolysis, optimization of storage of blood components, and innovative countermeasures for blood-eating parasites. A particular focus of it is, however, which I think we'll hear about today, is the development of a holy grail in the field of all of medicine, which is artificial blood substitutes. This has been a holy grail with all the good and bad connotations. It's more than 15 billion has been spent in this enterprise with many failures. So we're really hoping that this new approach will carry the day. He's actually told me a bit about this when I toured his labs, how he's mixing lipid nanoparticles with hemoglobin, but also in adding DPG, which is an allosteric effector. So this artificial hemoglobin in these small liposomes could actually respond to changes in oxygen tension with allosteric binding and release of oxygen. In 2016, Dr. co-founded Callicite Incorporated to commercialize Erythromere, this bio-inspired artificial red cell that I mentioned, and he's serving as Callicite's first president and is currently its chief scientific officer. And Callicite's been supported by nearly 10 million in federal grants and investor funding. And so we're absolutely delighted that he's moved here from St. Louis. And really, I've been incredibly impressed with your vision and leadership. He also has great ideas surrounding developed bioengineering here, which are something we're equally enthusiastic about. So without further ado, I'll introduce Dr. to the podium. Thank you, Allan, and congratulations.
Thank you, Mark. Just say it's been a thrill to work with you. Super excited about your leadership here at School of Medicine. And thank you for coming. And so what I'm going to tell you about is, how we've dealt with the challenges associated with creating fake blood. And there are many. And in fact, when I was given a questionnaire about what I thought the most important obstacles were to the development here. And after a little thought, I'd have to say that the most important obstacles really were my own sort of ignorance and inexperience in trying to deal with this problem. And so I've taken inspiration from nature. That's the bio-inspired design. So steal like an artist, as my wife says, or steal like a scientist from nature. And then also it's taken a pretty extensive team, as you'll see, to address all these challenges. And so I'm hoping, to lay all that out for you. Obviously, there's a disclosure here. I have intellectual property and equity in Callicite, and we'll be discussing that subject. So this is the outline for what I'm going to tell you, and hopefully spend a little more than 5 minutes on each topic. I'm going to focus on the unmet need, like why are we trying to do this? What are the design parameters for the perfect solution to this problem? Importantly, to understand the physiology of the system that we're working in, and I'll give you sort of a tongue-in-cheek example of what happens when you don't understand. The specific design solution we have conceived as a group, the approach to making it, how it works, the approach to dealing with the FDA, and then finally the pathway to commercializing it. So first, I want to acknowledge the absolutely fantastic team that mainly Elaine Haynes here, our CEO, has built in Kalasite. We've got a wonderful group just across the way working together. And these are the people that do really all the hard work. This is the board. And as you can see, they come with sort of a lot of connections. The original idea, in fact, came from my friend and partner in starting Kalasite, Dipanjan Pan, who is a synthetic chemist and bioengineer, who called me up about 12 years ago saying he had made a nanoparticle that looked a lot like a red blood cell. And did I think we could stuff hemoglobin in it, literally what he said, and, and make it work, because he knew about all the problems with the blood substitutes. He was building particles for imaging and was trying to make something with a big surface area relative to the volume. And as you'll see, what he made looks just like a red cell. So we fiddled around with it. It took 2 years to get something that would even start to work. We were able to get some small grant through which I met Dan Getman. So this was a really fantastic program at WashU where intellectual property that had therapeutic impact was funded. So rather than classic White Tower discovery science, but the scientist wasn't allowed to run off with the money alone. You're partnered with somebody who had formal R&D experience, and Dr. Getman had been the international vice president for R&D at Pfizer. And so he supervised me in the project, and I learned an incredible amount from Dan, who eventually joined the board of our company when we formed it. Dr. Spinella was another co-founder who has a lot of experience in transfusion medicine and clinical trials, and the three of us formed the company. Eventually recruited Dr. Elaine Haynes, who had a pharmacy background and had run a large division at Mallinckrodt before spinning that out and wanting to do something really fun, like making fake blood. And then we have two other, excellent board members. And, honestly, we get a lot, we meet very often, you have a very engaged group. And this is the scientific advisory board. My mentor and chair, Alan Schwartz at WashU, Greg Lanza, a giant in nanomedicine, Urshad Chowdhury, who's incredibly experienced in shock resuscitation immunology, Gene Moore, one of the giants in transfusion medicine in the setting of shock and trauma, Paul Ness, who's the editor of Transfusion and also a major figure in blood banking, and Steve Gould, who ran the most successful company that tried with the first generation. So again, a great team. And I can't emphasize enough how much we all rely on all these people. And the team is embedded in an awesome environment. And this is why we came, why Elaine was willing to come too. And you can see from this map here, all the resources we need to collaborate and to perform are nestled in one geospatial location with just, it's like being in Florence in the Renaissance. I mean, it's really fun place to be, couldn't be happier. And it's different than being in the middle of Missouri, just saying. So, so now on to the problem. And first, a little background and, a favorite subject, the history of transfusion. So if you're most of the time in medicine, if you think about it, doctors used to take blood out. I mean, they thought bloodletting was the way to go. Now we put it back in. So this is, the first major breakthrough is let's not take it out and put it back in. This followed sort of a pretty simple observation by a very smart guy who was playing with the veins on his forearm and noticed that the blood seemed to be flowing. And after sort of a new look at the way people, after cutting things up, created the concept of circulating blood. We used to think, medicine used to think blood was created in the liver every circulation and disappeared somewhere else. And that this idea of circulation was the first step. And then there were some attempts at transfusion, which were pretty weird. Literally putting blood from a lamb into a person to calm him down. Didn't work. And In fact, transfusion was outlawed for almost 200 years before the first human-to-human transfusion was done by an obstetrician who was sick of seeing pregnant women bleed to death. And the first transfusion was from a husband to his bleeding wife, a dedicated husband, okay? It basically just bled into a funnel that was taken into his wife, and luckily they were compatible in more ways than one. Since we didn't know about ABO matching back then. And then, as you can see, there were a series of improvements until the 1950s, and that's about it. So basically, we're using 1950s technology for transfusion. So what's the problem? So the problem that we're trying to deal with is that when you have to put blood in, you have to do it in a timely fashion. So we are aerobic machines, meaning we have to have oxygen continuously supplied by the cardiopulmonary system to every respiring cell, including the pump itself, the heart and the brain. And if you don't do that, you die pretty fast. And if you get injured and the blood runs out of the pumping system, your blood pressure gets too low, and it doesn't matter, if you can get to the hospital, there's injury, what we call the secondary injury from the trauma. And we can't get blood to the people at the scene of the accident because it's a living tissue and it has a cold chain and has to be preserved, and you have to cross match. And so it's just not practical. The fact is 30,000 people every year in the US bleed to death before they can get to the hospital. And there were about 1,000 combat fatalities in the recent conflict that would have been survivable if in-field transfusion had been available. In fact, this is the reason for all the majority of survivable casualties in the US military. So these are all preventable if we could just give them blood in the ambulance. Every single minute there's a delay, the mortality increases by 5%. Yet about 50 million people live more than 2 hours from a trauma center, and ambulances don't carry blood. This is a major public health problem. So what is the statement of need? So we need a carrier when blood is unavailable. We also need one when it's undesirable, but we can do it. This is uncross-matched blood, perfusion systems, etc., where blood kind of works, but it isn't as good as it could be. And then there are situations where stored blood is inadequate, civilian mass casualty events. Few people realize that the Boston Marathon bombing, they practically ran out of blood. They had to get blood from Maine in order to transfuse most of those people. If we're hearing about just in the news today, sadly, the possibility of a dirty bomb in Ukraine. Acute radiation syndrome is something that could happen if there's a nuclear event or a big nuclear accident. There will be a disruption in the power supply. There'll be a disruption in the supply of blood. In fact, when I presented this project at the American Society of Hematology, someone I had never met before came up to me and said, "Have you thought about what would happen to the blood banking system in nuclear war?" And I had to say, no. And she said, well, our agency is thinking about it and we're interested in this project. So there's a desire to create a depot for this type of thing so that we would be ready in those events. So many people have tried, as Dean Gladwin mentioned, and it hasn't worked. And I'm going to explain to you why. It doesn't carry oxygen the way it's supposed to, and it interferes with blood flow. And so you have to respect the physiologic system that you're trying to work within in order to develop a new therapeutic. Now, they failed in part because they didn't have the right tools. And those tools are now available with explosive improvements in synthetic chemistry. We can now etch your name in a nanoparticle with synthetic compounds. And you can also encapsulate biologics with programmable polymers and change the behavior of something that's toxic in plasma, guide it to a target, or change the way it circulates. And this has really made this sort of thing possible. So now we're trying to make an artificial red blood cell instead of a blood substitute. It's a different thing. What else could we do? we could, beyond just doing what red cells do, we could do more if we have control over it. Of course, we're trying to do the first thing first, but this is a therapeutic platform that has potential. So we could make red cells crafted to specific context. Our red cells adapt to altitude. We could pre-adapt the artificial red cells. We could disrupt sickle cell sequestration crises by delivering an intravascular oxygen supply to red cells that are locked away somewhere in the body. We could enable high-risk endovascular procedures by delivering oxygen therapy through the end of a catheter beyond a balloon. The limits to those procedures are you can't interrupt blood flow to the heart of the brain for more than a fixed period of time. But if you could give oxygen past the occlusion, you could do more. You could put pharmaceuticals on the shell. You could perfuse organs. You could perfuse an organ or tissue sewed into the body until it grew a new blood supply. So there's all kinds of new therapies that we could do if we could just get the basic thing to work. So before I get into that, I wanted to tell you about 3 people I admire greatly with some advice that I think is relevant for this project, or really for a lot of projects. We're going to start with Marilyn. So she was thinking about something else at the time, but the fact is that we have to be willing to embrace new ideas, and we have to be willing to take on more expansive, innovative concepts and look for new interpretation so that we can solve old problems. If something isn't working, try something new. So, somewhat different character, but what I'm talking about here is we have an incredibly adaptive system, and our system is designed to respond to stress, our human body. And it has controls that respond to stress. I'd say that somebody bleeding to death is under stress. And if we're going to intervene in that situation, we can't disrupt the normal controls that keep the body stable. So we have to understand the stress response, and in particular, the stress response in oxygen delivery. We'll talk about why the prior blood substitutes might not have worked. And finally, a relatively smart guy. We all want to keep things simple, but we can't make them too simple. We have to respect the complexity of the system. And so with that in mind, let's talk about parachutes. So we have to also test our therapeutics in the right way, or we can't really appreciate whether they're working or not. So nobody would argue whether these things work, but imagine if you tested it without realizing how high somebody had to be, you wouldn't be able to tell. You'd have people jumping out of a plane on the ground with a parachute and say, this isn't necessary. If you jumped before they had time for the parachute to open, but high enough to kill somebody, you'd think, well, it doesn't work, everybody's dead. If you jump them over water, okay, water breaks the fall and saves the people who don't have a parachute, but the people with the parachute drown because they get tangled up. Finally, you test it in the right way and you find out it works. So the design of the experiment revealed the benefit of the therapy because you understand the system. But if you just include all this stuff, you can't figure it out. So this is what we're up against. Honestly, it seems a little silly, but the fact is we don't really know if it's going to work or not. And we have to sort of force our way through these experiments and very thoughtfully consider if we're evaluating things in the right way. So what should we be doing if we want to design a blood substitute? Well, it has to be, we're starting with hemoglobin. There are other approaches, but let's begin with that. And we need it to be simple, and we have to have tunable components. It has to imitate the behavior of a red cell, and we're going to talk about that. And it can't interfere with other things that red cells do. It has to be easy to deliver. It basically has to be delivered by someone with a high school education who's frightened and in a stressful, difficult situation with limited tools. It has to be suitable for the intended use, which is to bridge someone from an accident or trauma scene to an emergency room. And it has to eventually be able to support something we call prolonged field care. If anybody saw Black Hawk Down, those poor kids who died, I should say young men who died, all bled to death. Every single one of them had a survivable injury, but they were caught for days and they couldn't get evacuated and they just simply bled to death. The medics had field transfusion, they wouldn't have died. And it has to be cleared without causing any problems. It has to be safe, sterile, can't interfere with the immune system. And importantly, it can't interfere with blood clotting. If you're getting this therapy, you can bet you're bleeding. So if you're bleeding and you interfere with the clotting, no bueno. So we can't do that either. It has to be compatible with other blood components and potential therapies that would be used. It has to be stable, and it can't interfere with lab instrumentation, kind of a tall order. What have people tried? There's a long, long menu, and I'm not going to be able to go through all of these, but people have tried to chemically modify hemoglobin in lots of different ways. They've tried to envelop it in different ways. And eventually we've gotten to super complex designs, and it's simplified a little bit more with this, what I'll call the top of the evolutionary tree, the artificial red cell. You can see why when Dr. Pan made these, he said, oh my goodness, I wonder if we can put hemoglobin in that, because that's exactly what red cells look like. So what happened with the first generation? And there were basically 5 of those chemically modified naked hemoglobins, I'll say, that were in clinical trials in the early 2000s. So this was all put together by a somewhat controversial but well-known intensivist, Chuck Natanson, that Mark Dean Gladwin used to work with. He takes all kinds of wild things on and put this together, and notice that basically the likelihood of death was higher if you got artificial blood. This is thousands of patients. And in particular, the likelihood of a heart attack was higher. And that was the same regardless of what type of patient, whether the control was a blood product or not, and whether you used any one of these. And as you can see, there were almost 4,000 patients in this analysis. And that meant you were more likely to die and you were more likely to get a heart attack. And this was, there was no learning in the system. So the rate of this problem was the same throughout the clinical trials. And the FDA looked at this and said, we're done. They closed down the entire field. As Dean Gladwin had said, nearly $15 billion went into this. And it went up in smoke. And as you can imagine, that dampened enthusiasm for investment in this space. So why did this happen? So we have to understand the physiology of the targeted system. And one thing to keep in mind is this little cell that looks super simple is the product of about a 400 million year forward genetic screen, natural selection. And the selective pressure there was stable O2 delivery under extreme stress, or you died before you could reproduce. And if you died before you could reproduce, that red cell didn't make it. So it had to work. You had to outrun a tiger while bleeding, basically. And if you could do that, you could reproduce, and that red cell, this is what came of that pressure. So we would be wise, to try to imitate it and not do something, innovative, like maybe put the hemoglobin in the bloodstream without a membrane around it. There's no organism more complicated than a worm that has unencapsulated hemoglobin. Something to think about. So in our body, we have about 20 to 30 trillion red cells. It's overwhelmingly the most numerous cell in the body. There's a significant portion of our nutritional and energy budget devoted to this task. We're making like 200 billion of these a day. So very conservative evolutionary pressure on where our nutritional and energy budget is spent. And if we invest this kind of resource into that little cell, it's important. And they have many, many roles beyond simple oxygen transport, and we have to understand that and respect it before we try to manipulate it. And the first thing to understand is this little molecule that was mentioned, 2,3-DPG. This is a tiny little remnant of glycolysis, okay, or metabolism of glucose that interacts with hemoglobin and changes the way it binds oxygen. So red cells have to do two opposite things. They have to go through the lung, and while going through the lung, capture oxygen. And then after they go through the lung, they come back to the heart, pumped out to the body, and let go of the oxygen. And chemically, that's opposite. So it will release the oxygen down an oxygen gradient, but that's not enough. We need to change the affinity as the red cell circulates, and it does it in other ways, but this is the thing that we exploit. So red cells have a more complicated system for doing this, called the Bohr and Haldane effect. They involve complicated chemistry and ion pumps that require energy and so on. We don't replicate that. Anyway, this is the way we measure it, through something called the oxygen dissociation curve. Here we plot the amount of oxygen. Here we plot the amount of oxygen bound to hemoglobin. When there's a lot of oxygen, a lot's bound to hemoglobin. When there's little oxygen, there's little bound. And this is the relationship with no DPG around. And progressively, as you add DPG, that curve shifts and the O2 affinity goes down. And so that's measured with something called the P50. That's the partial pressure of oxygen, where we have half saturation. And a shift to the right means the partial pressure needed to saturate's higher, the P50 is higher, and affinity is lower. It's convoluted like everything in medicine, but that's so higher P50 means lower affinity. That's important to remember. And our body does this. The molecule breathes in a way as it goes from lung to tissue, where it's inhaling the oxygen and exhaling the oxygen, and it's responding to these chemical cues that signal the adequacy of blood flow. So the chemical signature, when there's inadequate blood flow, pushes the curve to the right, and back in the lung, the chemical signature pushes it back to the left. So this is flexing back and forth during circulation, and it introduces this incredible amplification of the ability to move oxygen. So that's one feature. The other feature is the flow of blood. That's the flow of oxygen. So blood is carrying the red cells. So now we have the system. How do we distribute oxygen in this complicated system where we're actually only using about 5% of our ability right now? The tiger came and you had to run, you'd have to increase that by 100-fold. And so how can we do that? So perfusion lack must be sensed, flow must be redistributed, and it has to be effective in space and time. So how does that happen? So we've got this pretty complicated routing system, and this is the scalability in these elements that I'll talk about in a minute. And this pretty famous physiologist did a very simple experiment, and this was one of these sort of classic problems where they'd been doing things like jumping off the plane on the ground and thinking that the spinal cord controlled it. And eventually he designed the right experiment, flew the plane at 3,000 meters and figured it out. And what he did is he cannulated the femoral artery of a dog and put a flask of blood high and put oxygenated blood in the flask or deoxygenated blood, and then watched how fast it ran out. And when there was progressively less oxygen in the blood ran out of the flask faster, which meant there was less resistance to blood flow when there was less oxygen in the system. And the blood vessels dilated perfectly to stabilize the O2 delivery. Okay, as the amount of oxygen in the red cells went down, the flow increased perfectly until it reached the limit of the system, and then it tracked like this. And this was what he said. It's possible that red cells prevented accumulation of an inhibitor or released a factor that enhanced vascular responsiveness. This was in the 60s. And it took a while, and a lot of people like Dean Gladwin, in fact, to help figure out exactly what was going on here. And in fact, here's this very beautiful body of work. I encourage you to take a look at it. There's, many, many ideas about this specific chemistry, and I'm not going to get into it, but the bottom line is nitric oxide comes into red cells, magic happens, and it comes back out. in direct proportion to the amount of oxygen that's moving, so that red cells that enter blood vessels that don't get enough oxygen leave behind more nitric oxide than they encounter. And red cells that enter blood vessels that have too much oxygen leave behind less. So they're redistributing blood flow in proportion to need. And they do this on a millisecond time scale. And this is exactly what was going on in Guyton's dogs. And if we interfere with this, you will cause a heart attack. So if we interfere with this chemistry, which is going on in plasma, and you put a sink, then this prevents the physiology. Here, I'm just mentioning that it's not just enough that you have to have sort of the blood vessel open. We have to have the right amount of pressure in the blood vessel, and we can't interfere with the rheology. This is the viscosity of blood. So we have to respect the pressure gradient, the oncotic pressure, and the viscoelastic properties of blood. And all of those things have to be unperturbed for an oxygen therapeutic to work. And if you're wondering, here is forearm blood flow and oxygen delivery. 96% of the variation in oxygen delivery is explained by the difference in blood flow, and 13% is explained by the difference in oxygen content. Flow trumps content, so that we really have to focus on this and not that. And the first generation therapeutics focused on this at the expense of that, and that's why people died. So how do we get around that? So here were the flaws. The oxygen affinity was fixed. So we created a pseudo Bohr effect. We have this small molecule. Here is depicted as an artificial molecule called RSR13. We now use the natural molecule DPG. And basically there's an electrostatic interaction between this and this proprietary magic molecule that we put in the shell. We call this wetware. So this programs the behavior of the artificial red cell so that we now cue off the pH. So the pH in the lung is high, the pH in the periphery is low, and the pH changes this electrostatic interaction. So the electrostatic interaction is high when the particles are in the lung and low when they're in tissue. So this molecule is free or sequestered when the particle is circulating and the curve is shifting right, left, and the particle is breathing just like a red cell does. We built a barrier around the red cell to prevent or slow down the interaction with nitric oxide, so we don't interfere with that signaling. And we put in something to prevent the iron from rusting, and we lyophilize it so that it can be stored and reconstituted. And it's also engineered to be cleared in the liver, just like everything else in the body. And this is the chemical sort of pathway, again, from Dr. Pan, not me. This is a cartoon describing this proprietary molecule. There are other elements of the lipid. The magic is only about 5 to 10% of the membrane. This is all there for structure and biocompatibility. Other normal lipids from the membrane, plus this one that imitates the glycocalyx that's around a red cell. And this all breaks down so that metabolic byproducts are just like if you ate a burger. So there's nothing in here that ends up as a drug product. It's all designed to be broken down by endogenous molecules. So how do we make it? So this is called a swim lane diagram. We have inputs, a process, and outputs. And again, this is, a huge team is here developing this. This is something I get to talk about, but I didn't design this. So we start with mostly commercially available starting materials. We make the magic molecule, KC1003. Other lipids we make into a film, and I'll show you that. We mix it with the hemoglobin and the payload, and it magically assembles into an artificial cell because of the physical chemical properties of the lipids. And then this is then purified and then freeze-dried. And even the simple thing like purifying the hemoglobin is a huge process. And this is a little bit about how we make the liposomes. You take the lipid mixture and dry it into a film, then hydrate it with the payload, which is in water, It forms these little vesicles, okay, that then need to be sort of homogenized or made more uniform. And then they get purified and characterized. The way we purify it is through this membrane called, using a device called an extruder, which can go up to liters and liters. And we take these complicated things and make them fairly simple and uniform. And this is the way we clean it with a special system called the tangential flow filtration. And this is a bottle of artificial blood. So when we make it, we've got a mixture of the good stuff and the bad stuff. And we take the bad stuff and filter it out. And that's what we're left with. 99.9 or 99.6, I should say, % pure artificial red cells. There's a lot of work into freeze drying it. And here's a little movie showing you how quickly it works. So this is the freeze-dried artificial blood that we're just adding water to. And what we imagine is that there'd be a bag with a wet side and a dry side, and a medic could slap it and mix the wet and the dry, invert it a little bit like that, and presto, add water. Now that's ready for transfusion. We can mix it with any of the fluids that a medic is normally carrying. We have good fidelity from the pre-freeze dried to the post-freeze dried. We have a little bit of work to do improving the payload loss. And importantly, this is showing that the membrane property that prevents the problem with nitric oxide is not affected by lyophilization. But how well does it work? Here's the electron micrograph of the artificial red cells. You can see they look a lot like red cells, but this is 100 nanometers. Red cells are 7 microns. So a red cell is 70 times this little line. So these are a lot smaller than a red cell. Red cells circulate about 5 or 6 million per milliliter. These will probably be circulating about 1 times 10 to the 12th or 13th per milliliter. So very big difference. This is a surface charge and stability assessment. This is showing that the rheologic properties of blood are not affected by progressive addition of erythromer. This red line is the viscoelastic property of our natural blood. And this is additional doses of erythromer in simulated massive transfusion. And we match the viscosity behavior of blood exactly. And importantly, this is the important feature called the yield point. Our blood moves in pulsatile fashion throughout the circulation. So it slows down and almost stops in capillaries, and then it has to be restarted. And a significant portion of the work that the heart has to do is just reinitiate that movement. So the blood actually starts to become a little structured as it slows down. It's what's called the non-Newtonian fluid. And then you have to break that structure again. And the force required to break that structure is called the yield point. This red line is natural blood, progressive additions of erythromer, and we don't interfere with the yield point. This would be a major problem. So this is the those oxygen dissociation curves that I showed you before. And if you remember, shifting to the right is good. So these are normal red cells. This is erythromer. And you can see these are a little shifted to the right. And this is the way red cells move with pH, and this is the way erythromer moves with pH. And so we've imitated this behavior, and this is using RSR, but we also now use this other molecule, DPG. And this is showing the change in P50 as a function of pH. And this is showing free hemoglobin, which has a very low P50, meaning high affinity. And this is a natural red cell. These are stored red cells. So stored red cells aren't quite as good as natural ones. This is erythromer without any RSR, and this is erythromer when we load it up. So we can, in fact, make it even more potent than a red cell in terms of moving oxygen across the physiologic gradient. And if you index everything to stored blood, because that's what people might be getting, we can make it almost 4 times as potent, which means we can use almost a fourth of the same of hemoglobin. So that improves the safety because we can use a lower dose. What's the other key feature of the safety? I told you we don't want to interfere with nitric oxide, which is controlling vessel caliber. So this is a measure of nitric oxide consumption. Down is bad. So down shows nitric oxide being consumed. And this is a vessel reaction. Up is bad. Up is showing constriction. If you take that little window, the first few seconds, because this is a dynamic system, so blood is moving past a blood vessel. We just want to slow things down so we don't have have nitric oxide getting trapped. That's the trapping occurred by free hemoglobin, and erythromer is matching red cells. And if you do some funky math and look at the rate of change, you can see that the rate of this reaction is basically the same for erythromer and red cells, and the rate of the reaction in the blood vessel is the same. And in fact, they match. So we don't interfere with this problem. So what does that mean? So what we've done by increasing the oxygen flux is increase the potency. By reducing the NO trapping, we've reduced the toxicity, so we've broadened the therapeutic window. So this extends both the safety, extends the safety and makes it, improves the dose interval for us. And so by modeling all this, including that, we think that the dose interval is somewhere between 5 and maybe 8 to 9 hours, which is suitable for a prolonged field care situation. So now I want to show you how it works in an animal. And to do that, again, we wanted to think hard about the right experiment. We didn't want to do something easy, like jump off the plane on the ground. We didn't want to do something stupid, like jump over a swimming pool. We wanted to do it exactly the way that it works in the body. And so what's if you could figure out what molecule is the one you wanted to measure, the one that reports the balance between oxygen delivery and consumption is a molecule called HIF, or hypoxia-inducible factor. We've recently won the Nobel Prize, and this is a transcription factor that executes a genetic program that adapts the body to oxygen supply lack. So it tells us not just is someone hypoxic, it tells us each cell is physiologically relevant hypoxia. So there's somebody clever, not me, took a mouse and turned it into a firefly that would light up only when HIF was stabilized. So they fused luciferase to the oxygen-dependent domain of HIF-1 alpha. And if you give that mouse luciferin and take a picture, then you could count the photons and you could see how much HIF is in the body. And you can tell whether that mouse is getting enough oxygen or not and where. And if you make those mice anemic, they glow. In fact, they glow almost in direct proportion to the degree of anemia and its recovery. They glow and then it goes away. And this has been validated against, the protein and the mRNA. It's a very reliable system. We take these mice and we do an exchange transfusion and remove 2/3 of their red cells, they light up. And then we do a second exchange transfusion and replace that with artificial red cells. So now 2/3 of the hemoglobin is in erythromer, the light goes out. And we let the erythromer dissipate and the light comes back on. So this is a nice experiment. So this tests the entire system from lungs to heart to vascular tree to blood flow distribution. If we're interfering with anything in oxygen transport from air to cells, HIF lights up. And so while erythromer's in circulation, it's working, and while it's not, it's not. So we feel good about that. And this is just showing this in a more quantitative way. So now we're testing it in larger animals, and we've picked rabbits for a lot of reasons that I can go into later. But here we're simulating shock, and we take rabbits and we monitor them for a little while, and then we simulate a pretty big bleed, 40% of their blood volume. We take it out. We leave them down for a while to simulate the time it takes for a medic to get there. And then we resuscitate them with either blood or albumin. So this is an albumin-containing fluid that's like plasma, or erythromer, and then we watch them. And so they're a little bit different at the beginning, and now here they are in shock. And this is showing the mean arterial pressure, the amount of lactate. So lactate is an emission when you're burning glucose without oxygen. So when we burn glucose with oxygen, we just get carbon dioxide and water. And if we lose the oxygen, we can't complete the combustion of glucose, and you end up with lactate, and that accumulates in blood. And that's what causes the burn at the gym. So lactate is an important measure of anaerobic metabolism. And then this is measuring PO2 in the liver directly. So we've got direct, indirect, and the blood pressure. We normalize everything to the last moment. The animals were all the same, and this is blood, erythema, albumin, blood, erythema, albumin, and we come close to normalizing the blood pressure. It's basically the same an hour after. And then we get rid of the lactate and we bring the PO2 normal. And I want to show you now some survival experiments. So now we're testing it to see if it's going to work to save a life. And so we take these rabbits, and 2 days before the experiment, we instrument them with a central line so we can access their blood vessels. We bring it out to a button. And then we put in a telemetry device. It's a little button battery on a fiber and a Bluetooth transmitter, and this is a quarter. And that's in their femoral artery. So we can monitor their heart rate and blood pressure. We do this so we can do this under very light anesthesia, because otherwise they won't tolerate this massive thing we do, which is take out half their blood. So we then take out half their blood in a very specific schedule and leave them down. Now we're doing this for an hour. And then we resuscitate them and monitor them. And if they look like they're about to die, we euthanize them. And this is sort of what the amount of blood that we take out of a 2-kilo rabbit. And this is a rabbit lightly anesthetized, getting resuscitated with erythema at the beginning. Now he's getting his second dose. And now he's had his fourth dose. And literally we watch them wake up while they're getting it. And this is the blood heart rate pattern and blood pressure. You can see it's very different. We're now comparing it to salt water. And this is where we are with the survival analysis. This is a survival in this model with blood. This is a survival with salt water. We designed this to be what we call an LD50 model. And now we've got survival with erythema, and it's very close to being significant over salt water, and hopefully will remain not different from blood. And we're close to finishing these experiments. So now what do? Our task is to get to the point where we can test this in humans. And we've had a pre-IND meeting that Lane led with the FDA a little while ago. They mainly blessed our approach. We've got a few things that we need to tweak, and now we have an outline for a clinical trial program and an approach. Beyond that, so these are the things we've done so far. These are the things we have left to do. It's a lot. We have to scale, we have production, we have to get the toxicology done, a large animal safety pharmacology. We have to make it under a special good manufacturing practice. We have to package it. We have to do the clinical trial program and so on. So there's plenty to do here, but we've done a reasonable thing so far. This is our development plan and funding. It has not been cheap. And we have been fortunate to have a significant government investment from multiple agencies into the program. And we have a large piece of that DARPA award will go towards erythema, and then several other grants that we expect to get funded in the new year. And we are closing maybe sometime soon on a new raise that will help us get through to phase 1 trials. This is the clinical trial design. We have several multiple phase program culminating in a pre-hospital trauma trial that we think will take about 600 subjects. And as I mentioned, expect we've probably or hopefully will be in humans in mid to 2024, and maybe by 2028 or 2030, you can buy it at your local drugstore. So I do want to acknowledge this incredible team, both in the lab and all over the country, in fact, the world, and the outstanding KLSI team and advisory board, and without whom none of this is possible. So thank you for your attention, and happy to answer any questions.
Fantastic presentation. Thank you. Why are you switching from RS R13 to DPG? Or are you planning to have different sort of tunable P50s? And one of the challenges, obviously, of artificial blood is acute kidney injury. Have you looked at whether any of the hemoglobin that might come out of the polymer gets into the kidney?
Okay, there's a little in there. So why are we switching? So there's no. It's not at all. So we started working with it because it's a little more potent than 2,3-DPG, and it looked a little easier to shuttle in terms of the chemistry. What we find though is that there's a feature called the log P, which tells you whether a molecule is likely or not to cross a membrane or a lipid membrane. And unfortunately, RSR has a disfavorable log P, meaning it will go in and out of a membrane and it goes out, it leaks. So once we put it in the body, it ends up in plasma and then it ends up in red blood cells. And so we end up actually dosing the recipient red cells with the RSR. One of our projects is going to be to tweak the RSR structure to keep it inside, but we don't want to slow down our development. And so we've switched back to DPG, which because of its, it stays in red cells, as we know, but because it stays in our red cells, and it will stay in erythromer. And it works almost as well enough for us right now. So we think we can modify RSR so that it'll still work and keep it inside. And then we will have the tunable system where we would have a little bit of either probably. The other feature is that RSR is a drug substance. DPG is natural and would be considered GRAS or generally regarded as safe. So it's a little more pragmatic, and it also is a regulatory, sort of removes a little bit of a regulatory issue. As far as the kidney goes, that's a huge problem, as you know. We have monitored, we are about to start some experiments where we monitor GFR in these resuscitation systems. We haven't done it yet, but we also know that none of the hemoglobin ends up in the urine. It all ends up in the bile. So I haven't shown any of the biodistribution or elimination data, but erythromer is cleared by the mononuclear phagocytic system in the liver. It escapes the spleen and gets caught in the liver, and it's too big to get filtered in the kidney. And very little of it escapes sort of the particle. So the particles get cleared before they degrade. So right now, the timing of all those things is working well. One possibility, though, is that the clearance system saturates in a massive transfusion, and then the particles would circulate for an extended period of time. And it's possible that they might break down, but we haven't, there's always another hill to climb, and that may be one, but we haven't tackled that yet.
This is really cool. Sort of an annoying question. What sort of a time constant do you enjoy with that IF1 alpha luciferase fusion protein?
Yeah, so it depends on circumstances. So if the so you're talking about how fast does the stabilized construct clear if you normalize oxygen delivery? Is that what?
Response time to changes in oxygen tension.
How quickly does it? It's as quickly as HIF. So we can see a pretty quick response. So it takes us the time it takes us to do the intervention, get the animal over to the camera, set everything up, and do it all. We've never sort of seen the response evolve. So it happens within the time scale of this walk. We haven't done sort of experiments in the camera, where we manipulate the amount of oxygen to see, how fast it occurs. But the timing works pretty well for us, and it's stable as long as you don't normalize the O2 delivery. once you normalize O2 delivery, the signal goes away pretty quickly as HIF goes away. Yep. Yes. It's not. So, there is a very tiny amount that as you probably know, that gets bound to hemoglobin. But that is, because we're using so little, in fact, that's physiologically irrelevant. So there is a chance that carbon dioxide will accumulate. Now, why will that matter? So there's one circumstance. So if you don't have brain injury, that might actually be a good thing, because hypercapnia or mild hypercapnia actually enhances the type of vascular responses that we're looking for. And in fact, we sometimes do that on purpose, or as Dr. Glavin does in the ICU. So we allow the CO2 to creep up a little bit. And in fact, that improves gas exchange in the lung and can improve oxygen perfusion matching in the periphery. However, if you have a brain injury, it's not good. So CO2 accumulation will amplify intracranial hypertension. So patients who have bleeding and closed head injury, the accumulation of carbon dioxide might be a problem. So that's not something we can do anything about without adding something else to the construct, which would be adding carbonic anhydrase and possibly something else to help get rid of the carbon dioxide. But we're trying to do it seems complicated, but what I'm calling the simple formulation, which wouldn't take care of CO2, but it's an insightful question, and will be relevant with, and there are a lot of people with the brain injury. Now, those are people who will hopefully survive to deal with their brain injury rather than bleed to death. Yeah. Yes, you're absolutely right. So it will possibly change, ventilatory control, and that, as you know, changes the threshold for, yes. So if somebody gets a little too hypercapnic, they'll slow their breathing down, and that won't be good either. So.
Well, thank you, Allan. That was Thank you for describing your work in such a wonderful detail, but also just amazing, the big science that you're undertaking. And I've got to say, having spent some time with you, these are not just names on the screen. You do a wonderful job orchestrating and involving and bringing in expertise from around the country. And certainly the team you've built here is amazing, and how quickly you've become embedded within the University of Maryland. So thank you. Thank you. Would like to, the next part of the program is many faculty at the university work hard and are awarded patents, which is not an easy thing to be done. So let me ask Phil Rogalato to come up and tell us about the patents that are awarded this past year. And we're going to hand out some plaques with the patents.
Thank you, Jim. It is great to be able to do this in person again. So yeah, as Jim said, we're gonna, like we do every year, we're gonna go through a list of inventors on new US patents that issued during the past year. But I do want to take just 10 seconds, 15 seconds and acknowledge all of our other inventors that aren't on this list. Because if you think about it, we have between new US provisionals, divisionals, continuations, new foreign patents, we probably have every year over 200 new filings. And this year is no different. We had 235. If you add that to the 130 new invention disclosures, that is a lot of very inventive activity here at UMB. So congratulations to everybody, and thanks to all of the inventors on campus who take the time to work with us and try to protect their ideas in the form of a copyright or a patent. So with that, I'm going to read through this list. And I remember from doing this previously, it's really dark in this corner. So We're going to go in alphabetical order by school. So we'll go School of Dentistry, and then Medicine, and then Pharmacy. And if I call your name, and I'll call any of the co-inventors that might also be here, you can come on up and get your plaque from, I guess, Gail or Jim, and have your picture taken to kind of commemorate this, this issued patent. So first up, we'll start with School of Dentistry. And the first inventor is Dr. Bob Ernst. All right, Dr. Ernst is not here. He is, of course, a previous Entrepreneur of the Year. Next up is Dr. Hanpin Fang. Okay. Next is Dr. Roddy Masri. All right. And then last on the School of Dentistry list is Dr. Henning Wren. All right, that is a shame because that is a very cool technology. All right, so we're going to School of Medicine. And the first, actually 3 patents are Dr. Kurt Sivens. He's not going to be here. He already told us he wasn't. He already got his patents, or sorry, his plaques. So next is Dr. Richard Eckert. All right. I never finish on time. Perhaps I will today. All right, so next on School of Medicine, we have Dr. Dawei Gein. Okay, and none of his co-inventors actually have moved on. Dr. Bingren Hugh. All right. All right, next up, I know he's here. All right. Dr. Peter Hugh and co-inventors, Dr. Deb Stein, Colin McKenzie, Lynn Stansberry, Tom Scalia, and Shiming Yang actually has 2 patents. Method and apparatus for predicting need for blood transfusion, and method and apparatus for monitoring collection of physiological patient data. 2 very excellent inventions with a lot of commercial interest. Thank you, Dr. Hugh. Next up, I don't know if these folks are here. It's Dr. Bart Griffith and Dr. Zach Kahn, Chetan Pazura, Murdad Qureshi, and Greg Biddle, also from from UMMC, we're residents and fellows. This invention, of course, Dr. Griffith is also a previous Entrepreneur of the Year, and this invention is an eConnect One, a new startup company.
I don't think we told people that if you don't show up,
you don't get your plaque. Yes, exactly. They just call. All right, Dr. Joe Lakowitz. I don't see him, but and his co-inventors, Dr. Al Reese and Dr. Badagu. All right. All right. I know this team is here. A great team. Dr. Stuart Martin. With co-inventor, Dr. Rebecca Bettis. I know she's here too. And their invention is materials and methods for assaying living cells. This technology was previously licensed to Self, a startup company out of UMB. It has come back to the university, a very promising technology. We are actively marketing it right now. Thank you. All right. All right. Next up is Dr. Kamal Mudgil. All right. For central nervous system homing peptides and uses thereof. And I'm glad you're here because this is a great invention. It was an MII winner. Several years ago, Dr. Mudgil was a presenter at the UMB Johns Hopkins Alliance meeting. It was very well received there. And this technology has now been licensed to a new startup, Silo Pharmaceuticals, and they are sponsoring research in his lab. All right. Next up is Dr. Anindo Roy. All right. And Dr. Richard Macco, co-inventors. This technology has been, of course, licensed to Next Step. It's method and apparatus for providing economical portable deficit adjusted adaptive assistance during movement. It is the subject of Next Step Robotics, which has been a UM Ventures investment company and a momentum fund investment company, doing very, very well. Next is Dr. Mohammed Sajadi. I don't know if he's here or any of his co-inventors, Dr. DeVico or Dr. Lewis. All right. Next is Dr. Charles Sanser. Very innovative surgeon inventor, but I guess he is not here. Next up, we have Dr. Richard Thompson, along with Dr. Zhang, and I don't know if anybody else is. Dr. Szymanski is no longer here. For methods for detecting and/or predicting age-related macular degeneration and/or Alzheimer's disease. This is Dr. Thompson is extremely inventive. We have had many disclosures and many patents over the years from him. And this particular technology is actively being partnered right now with a manufacturing company and hopefully with a pharma company as well. Congratulations. All right. The next person I don't think I saw, but Dr. Steve Trapello. Of course, his technology is the basis for highly successful UMB startup Coaptech, also doing very well. Dr. Jeff Wolf, another very innovative surgeon and inventor. His technology is the basis for UMB startup Sonosa. All right, Dr. John Wu and Dr. Bart Griffith. All right, that is a blood oxygenator technology, which is of course part of Breathe and now part of Abiomed after purchase 2 years ago. All right, so that was the last one in the School of Medicine. I just have a couple now from the School of Pharmacy. Dr. Paul Shapiro. And co-inventors, Dr. Jeff Hasday, if he's here, Steve Fletcher, and Alex McCarroll. They actually have two inventions. It's non-ATP catalytic site P38 mitogen activated protein kinase inhibitors. Both of these patents have been licensed to Genie, which is another UMB startup company doing extremely well, just completed a phase 1 study and is raising capital to start a phase 2. Very successful company located out in California. And last but not least, School of Pharmacy, we have Dr. Bruce Yu and Dr. Mark Caravan. They have the last two patents. I'm sorry that they're not here because after working with those two for quite some time, we just licensed that technology to a very large international manufacturing company. So anyway, thank you and congratulations to everybody. And I guess, you know, Jim will be up next to put together some closing remarks.
Just in closing, thank you all for being here. Thank you, Allan, for a marvelous presentation and all the work that you're doing. We have a reception out the door and down to the right, so please stay and mingle. And I think the dean has some more questions for you.